One of the environmental factors to which bacteria and other microorganisms are quite sensitive is the presence of oxygen. For example, some microorganisms will grow only in the presence of oxygen and are called obligate aerobes. Facultative anaerobes will grow either in the presence or in the absence of oxygen, but better in its presence. Strict or obligate anaerobes will grow only in the absence of oxygen and are actually harmed by its presence. We also have aerotolerant anaerobes, which are microorganisms that cannot use oxygen but are not harmed by it either. And finally, microorganisms that require a small amount of oxygen for normal growth but are inhibited by oxygen at normal atmospheric tension are called microaerophlies. So these are the different classifications of bacteria based on their oxygen requirement. Remember the keyword for obligate aerobes, so presence of oxygen, facultative anaerobes, presence or absence of oxygen, obligate anaerobes, absence of oxygen, aerotolerant, so they cannot use oxygen and are not harmed by its presence. Microaerophiles require small amount of oxygen. These variations in oxygen requirements can be easily seen by inoculating a tube of molten agar with the bacterium in question, mixing the agar thoroughly without aerating it, and allowing it to solidify. The bacteria will grow in the part of the agar dip culture that contains the proper oxygen requirement. As you can see, these classifications of bacteria based on their oxygen requirement have different growth pattern on a molten agar. The damaging effects of oxygen on anaerobic bacteria create difficult culturing problems. Ideally, one should not provide an oxygen-free environment, but one that has adequate amount of moisture for bacterial growth. It is also necessary to have carbon dioxide present for the growth of many anaerobic bacteria. There are a number of ways in which anaerobic bacteria may be cultured. So one way to culture anaerobic bacteria is to use a anaerobic gas chamber. So this is not present in all laboratories. Some laboratories do not have this kind of equipment. So the three most useful will be described later. One of the most convenient approaches is to employ specially designed commercial anaerobic broth. Two of the most useful are cooked meat medium, so the one on the left side, and thioglycolate broth, which can be purchased with methylene blue or visazurin as an oxidation reduction indicator. When this medium begins to turn bluish or reddish, it is becoming too aerobic for the culture of anaerobic bacteria.